welcome to lecture number 2, and we'll be talking about NMR-based metabolomics and some of the methods and protocols. NMR spectroscopy involves a magnet and a computer and radio frequency electronics. And typically what you're doing is you're collecting a spectrum that looks not unlike the spectrum we saw for HPLC and GC. There's just lots of peaks. The peaks are not separated by time. They're separated by frequency. And these represent the resonant frequencies at which nuclei are, if you want, oscillating or vibrating. So if you're explaining NMR to someone who's never had it or seen it before, you basically have to describe it this way, in that you're putting a sample under a strong magnetic field. And that magnetic field basically conditions the sample so that it's receptive to electromagnetic radiation at the radio frequency. If you don't put a magnet or anything on a magnetic field, it's not going to be susceptible to that EM radiation. So if you send in some electromagnetic pulses, radio waves typically, that sample will absorb some of that electromagnetic radiation. And that absorbance is not unlike the absorbance when we measure UV absorption or light absorption. And we can think of that spectrum of absorption not unlike a colored spectrum of light, where there's high frequencies and low frequencies, or long wavelengths and short wavelengths. And that absorption essentially is characterized by absorption bands. And we call those absorption bands resonances. And that's partly the way we describe it, or physicists have typically thought of NMR as an optimal resonance. But I like to think of it essentially as these are absorption bands, where EM radio frequency radiation is being absorbed. And the absorptions produce very sharp peaks. So that's a simple explanation of NMR. It's also important to understand that NMR needs magnets, and the bigger the magnets, the better. And so as you increase the magnetic field, you'll have generally higher frequency absorbance. And you can also change with the different types of nuclei, and different nuclei will also absorb at different frequencies. So whether it's, again, high frequency, low frequency, high magnetic field strengths, low magnetic field strengths, these are critical to NMR. And we typically talk about magnets in NMR not by the strength in terms of Gauss or Tesla. We typically say this is a 500 megahertz or a 900 megahertz NMR, which is an indication of the strength of the magnetic field and the characteristic frequency of that magnet. So you've seen this picture before. Most NMR instruments are characterized by a magnet, superconducting magnet that sits on big legs. The magnet's about the size of a refrigerator. The samples can be either dropped in test tubes, or there are flow injection systems where you can suck the sample out and inject it directly into a flow cell. From the flow cell, the sample is exposed, or the NMR tube, to these radio frequency waves. You have both a transmitter and a receiver, so it's a giant-sized radio, basically what's beside the NMR magnet. It transmits the radio frequency, measures the absorbance, and then the absorption signal is transmitted to a computer, which will do various tricks to make the spectrum look nice. Almost all NMR magnets are superconducting magnets now. Means they're bathed in liquid helium and in liquid nitrogen. This is a cutaway view of an NMR magnet. There are a few places around the world where people have spent the time and money. But they're a giant thermos bottle, really, with a relatively small magnet. The magnet is made up of niobium tin alloy. It's wrapped around to essentially create an electromagnet, which is charged just once. And then it kept in very cold conditions, about 4 degrees Kelvin, so it stays superconducting. So the trick with keeping an NMR going is always keeping the liquid helium and the liquid nitrogen supplies around the magnet constant. There's an inner core, which is filled with liquid helium, and an outer core, which is filled with liquid nitrogen. And inside that, they use space blankets, which are sort of insulation and vacuum seals. So this is, again, sort of a cutaway view if you were to look at the position of where the vacuum lines are, which are in black, the liquid helium, which is in green, and then the liquid nitrogen, all trying to make things as cool as possible. So that's that overhead view on your left, and then on the right is, again, sort of a side view, a cutaway view. Now, in the center of the magnet is a bore, a hole. These are hollow magnets, or ring magnets if you want, with it's a coil wrapped around them. And under that hole, you will insert a probe, and the probe consists of a lot of the electronics that allow you to both send and receive radio signals or radio frequency signals. Magnets weigh a lot, so generally you have to mount them on magnets that are magnet legs that have special pneumatic pads to keep things steady. So an NMR probe is something that might be 50 centimeters long, again with lots of electronics in the base, resistors and capacitors that allow you to tune the coil. Inside the probe is a coil. It's typically called a saddle coil, and it's the thing that allows you to both send and receive radio waves into the sample. There's different geometries. The favored one is saddle coils, but many people have come up with better geometries that are slightly different. The saddle coil allows you to produce an orthogonal magnetic field, one in this case that's pointing to your right, which is a purple, whereas the main magnet, which is measured in tens of Tesla or hundreds of thousands of Gauss, is typically aligned with the z-axis in a vertical. And so it's the coil that allows you to do pulsing, and it's the coil that allows to measure the readout from the sample, which sits in the coil. So in NMR, we're mostly working with liquids, and the liquids are housed in tiny little tubes. They're about as thin as a pencil, in these cases often thinner, down to about 3 millimeters is a common sample size these days. So you'll put anywhere between 100 microliters to 500 microliters in one of these thin, pencil-thin NMR tubes and drop it into the spectrometer and start collecting. So superconducting magnets made NMR much more powerful, much more useful, much more stable. Another critical development that happened in the 1960s that earned a Nobel Prize was the idea of Fourier transform NMR, which allows you to basically excite all of the signals, all of the components that are resonant at the same time. And people have made it sort of the equivalent of like trying to ring a whole bunch of bells at once and listening to all the bells ringing, or listening to a whole bunch of people in a room chatting and then tracking all of their voices in a voice readout. Anyways, what you'll see typically with an NMR signal, if it's excited and if there's a sample inside, is you'll see a decaying oscillation. Just like if you ring a bell or a bunch of bells, they'll diminish over time in terms of their intensity. You can take that signal and Fourier transform it. So you're taking a time-decaying signal, and with Fourier transform, you're converting it from time to the frequency domain, or an inverse time. By converting it or frequency, you're actually able to detect the most significant frequencies that are in that free induction decay. And in this case, there are 3 significant frequencies, and these are plotted in the sample or signal below. So we're able to take, with Fourier transform, a signal that is exciting everything all at once, as listening to a crowded room, and then picking out all of the voices in that room, or ringing a whole bunch of bells and identifying which frequencies those bells are ringing at, or pressing all the keys on the piano and identifying which keys or notes were pressed. So that's the analogy with sound. This is done with radio waves. And this is an understandable spectrum. This isn't. In the old days, we did continuous wave analysis, and we would scan through the frequencies by changing the magnetic field and measure where we could get these absorbance frequencies. That would take a long time. Fourier transformation allows you to do it in seconds. So it sped up NMR tremendously and allowed people to get tremendous signal-to-noise improvements. So improved magnets, along with improved electronics and computers, made NMR in the 1960s really feasible and essentially the darling of the chemistry community. So every NMR spectrum collected in liquid will exhibit chemical shifts, exhibit certain splitting patterns, and they'll exhibit certain intensities. And the chemical shifts are the mileposts of NMR. This sample has 2 basic chemical shifts, and these chemical shifts corresponding to different protons or hydrogen atoms are split. And the splitting is caused by spin coupling. This is a doublet, this is a triplet. You can have quartets and so on. And this splitting is typically measured in a few hertz, whereas the chemical shifts are measured in a few hundred hertz, and we typically quote things in terms of parts per million. The intensity of peaks also allows us to quantify things. It tells us how many hydrogens there are in a given chemical, but also if we have an added sample that we know the exact concentration, we can also measure the concentrations via NMR very accurately. If you've done a bit of NMR in chemistry, at some point you've had to memorize something like this. These are tables of characteristic chemical shifts. And chemical shifts are determined largely by the electronegativity of nearby atoms in the molecule. And so depending on the electronegativity or the presence of whether things are saturated or unsaturated or oxygens or nitrogens or aromatic groups, you'll see very distinct chemical shifts. And if you've got this memorized, it is actually possible to look at an NMR spectrum and reasonably accurate figure out what the components are and what the molecule looks like. So the chemical shifts help in one regard. The coupling constants help in another regard. And so this is an example of bromoethane. And we're seeing how, again, having an electronegative atom like bromine causes a distinct shift, moving the hydrogen atoms to this particular chemical shift about 3.8. And then further down the molecule, which is still physically located further away from the bromine, we have these B hydrogens, or B protons, which are around about 2 parts per million. And because of the presence of 3 hydrogens or 2 hydrogens, we'll get different coupling patterns. So the 3 hydrogens on B produce 3 + 1 peaks. The 2 hydrogens on A produce 2 + 1 peaks for B. So this is the coupling, and it allows you to figure out which atoms are which and to design or determine what the structure is. So that's an NMR spectrum of a pure compound, and this is what we do in nanochemistry to figure out the structure of pure compounds. But what we do in metabolomics is we look at NMR of mixtures. And so those aren't particularly simple. There are lots of peaks there, and in fact, figuring out what the chemical composition of a mixture using standard 1D NMR is not trivial. And we'll talk about how that's done. So our focus in NMR-based metabolomics is looking at primarily mixtures as opposed to pure compounds, but still trying to deconvolute the chemical shifts and coupling patterns to figure out what really is there. So I'm going to take a brief tour on the history of NMR-based metabolomics and maybe give you some context. So the idea of using NMR to measure metabolites and even looking at mixtures was started back in 1974. And it was published in a paper by Wilson and Burlingame. Burlingame is actually a mass spectroscopist now, but he started, if you want, launched the whole field of NMR-based metabolomics. Bob Shulman did a lot of studies on NMR and metabolism in cell systems. And in many respects, he helped pioneer the general application because he did so many consistent and important studies showing that both proton, carbon, and phosphorus NMR were really useful in understanding metabolism. I had a chance to work with Bob Shulman back in those early years, and I guess that's where I first cut my teeth on NMR and also realized that you could use NMR for metabolite characterization. In 1984, Jeremy Nicholson, who's sort of the godfather of metabolomics, did the first characterization by NMR of urine, which at that time looked like a total mess. But he published it and commented how interesting or potentially interesting it could be. Another study came out and noticed that NMR could be used potentially to detect cancer in blood samples. And this generated a lot of excitement in the 80s, and then was largely shown to be incorrect, which sort of soured lots of people on NMR and metabolomics for many years. Picking up on the initial discoveries of fossil, Jim Otvos looked at blood and NMR and noticed that it wasn't really good at detecting cancer, but it was really good at detecting HDL and LDL and VLDL. And he developed a company called Liposcience that he sold for $85 million a few years back. In the early 1990s, a Canadian scientist was looking at MRI, or Spectra, in the early days of chemical shift imaging, and realized that you could actually deconvolve NMR spectra and could actually detect and quantify a number of metabolites. And so this led to a software tool called LCModel, which was largely ignored by the NMR spectroscopy community for 20 years. Because I guess MRI people don't talk with NMR people. In 1999, the term Metabolomics was coined by Jeremy Nicholson. And in 2000, the word metabolomics was coined by Drysdale. And for about 10 years, there was a battle between the two of what you should call it. The word metabolomics won. We started doing automated NMR of urine back in 2001. And it worked reasonably well, but it couldn't cover very many metabolites. It was very good at identifying inborn errors of metabolism. Other approaches were developed by Raphael Buschweiler to do covariance NMR and mixture deconvolution. Statistical spectroscopy started being developed by Jeremy Nicholson. And then a large number of metabolome projects based on NMR were developed or activated in Wisconsin by John Markley, and then by our group in Edmonton. So, as I say, the four key players in the early days: Al Burlingame, who's now a mass spec person; Bob Schulman, who's just celebrated his 91st birthday; Jim Otvos, who's a millionaire; and Jeremy Nicholson, who's world famous. So, as I said, the Otvos application was this idea of taking the NMR spectrum of serum and deconvoluting a specific signal around about 0.7 to about 0.85 or 0.9 ppm, and using the known signals of HDL, LDL, and VLDL to figure out how much was there. And you can see this is one person, and this is another person, and they have different levels. One has very high levels of VLDL. Another one has very high levels of LDL. So you can even measure many other sub-components of lipoproteins. And in fact, it's up to about 15 or 16 that can be routinely measured. So this technique has become highly used in the US. It's FDA approved. 1.5 million samples are processed every year by NMR. It's much faster than most other techniques and much more accurate. Chemical shift imaging, or MRS, again, this is a development where you can look at the NMR spectra of samples in the brain or in tissues, and believe it or not, that is an NMR spectrum. It's a lower resolution, but you can deconvolve that and identify specific metabolites in there, about 10 or 15. And now with newer developments, you're able to get some strikingly good spectra with higher field MRI instruments. So in terms of modern NMR-based metabolomics, the sort of practice, stuff that's practiced around by most NMR labs doing this, there are these two different approaches. The one largely pioneered by Jeremy Nicholson's group, which is to use statistics and chemometrics to identify, in this case, the red peaks as being the most significantly altered and the blue peaks as being the least significantly altered. So this was done in the days when we really didn't know what was in biological samples, when we didn't know what was in urine, we didn't know what was in blood, and we didn't know what was in cell extracts or saliva. But since then, a lot has been done, and over the last 5 years, much is known. In fact, we know almost all of the compounds that you can measure by NMR that could be detectable by NMR in blood, in serum, in urine, in saliva, in cerebral spinal fluid, in cell extracts, and in many juices and wines, and the list goes on. And so when you have a list of what you should expect and what's known, then you can start doing something else, and this is called deconvolution. And that means identifying all of the peaks and quantifying everything. So in this regard, even Jeremy Nicholson has gone from this to this, because they realized that there's no point if you don't have to do this. In those days, it was a matter of then once you found that the yellow-orange peaks were changed, what are they? And that becomes really difficult to figure that out. I'll talk about both techniques in detail in, I think, third and fourth lectures of today. So in terms of NMR-based metabolomics, as I showed with the workflow before, you start with a sample. If you need to, you can extract it. In all cases, or almost all cases, you like to look with liquids. It is possible to do solid-state NMR-based metabolomics. You can put solids in, whether it's tissues, frozen tissues. So with the magic angle sample spinning, you can get remarkably good spectra. People are doing metabolomics of soil. So it is possible to do NMR-based metabolomics of solids, but 99% of what's published today is actually on liquids, and NMR is particularly good at liquids, and typically what you'll do is that sort of data analysis. If you look at the detail, typically at how an NMR workflow is done, and I highlighted this earlier, is that there is typically sample preparation, where it might be that you're weighing things out or measuring things out, adding your DSS or TSP. And it might take anywhere from 20 to 30 minutes for each sample. If you've got robots or if you're really skilled, you can do this in less time. Typically, there's a sample loading and spectral acquisition for between 10 and 15 minutes if you've got a robotic system, or if you've got graduate students who are willing to work all day and all night. Spectral processing is relatively quick. You might have to do a bit of phasing. You might have to do some referencing. Most people, if they've done NMR, can do this relatively quickly. If you're new, it can take about 10 times longer. So these processes, they can be shortened to some extent, but the thing that takes the most time is actually that deconvolution step, which is why you're trying to figure out all those peaks, what they are, what they mean, how big they are, how small they are. And then even after that, there's the data interpretation, which can take anywhere from 60 minutes to 6,000 minutes. Depends on how far you want to go. So NMR, we highlighted other technologies in the first lecture, but NMR has a lot of advantages. And some of you who saw my earlier lectures probably saw this slide as well, so I'll apologize for any repetition. But for those of you who haven't, I think NMR is a very useful technology. NMR instruments are incredibly stable. The typically lifespan for an NMR is about 20 years. And there's very little downtime that you have. The magnets are stable, the technology is stable, the computers are stable. The only thing that might burn out over time are probes. Compared to mass spec, often if things go down once every month or two, you're considered lucky. They are, if you want, dirty instruments and have to be regularly cleaned and regularly tuned. NMR is a non-destructive technique. So it doesn't wreck materials. You can remeasure the same sample over and over and over again. Nothing disappears, nothing is burned up. In contrast with GCMS, LCMS, or any other technology, those samples are lost. They are destroyed as part of the analysis process. NMR is highly quantitative. And we talked about that with the dependence on the number of protons and the fact that you can scale very accurately. There's very little sample preparation. There's no HPLC, UPLC requirements. You can literally drop samples into the NMR tube. I've heard of people even peeing into the NMR. And you can start collecting. So it's a very, very minimal sample preparation. You don't have to derivatize things. You don't have to do any chemistry. You can detect a lot of samples that are compounds that cannot be detected by mass spec. Sugars are an example. Other non-ionizable compounds like alcohols are easily detected, polyols. You can measure living systems in NMR. You can't do that with mass spec. So with living systems, you can measure flux, you can measure kinetics, you can image, and you can see these things happening in real time. That's a significant advantage of NMR. And then when it comes to identifying those millions of unknown compounds, that dark matter that we talked about, NMR is the standard. It's how we identify things. Mass spec helps, but NMR is the thing that cinches it, and it's required for any publications these days. In terms of the popularity for NMR-based metabolomics, it's growing. It's growing exponentially. From a few publications in 1999 to 420 this past year and about 600 for 2016. That's what the projection is. So relative to other fields in NMR, NMR-based metabolomics is the most rapidly growing. If you look at NMR and proteins, NMR and some aspects of imaging, it's relatively flat or in some cases even declining. So this is a hot area. NMR doesn't allow you to see everything. And in some cases, that's actually good. There are different biofluids that have different levels of complexity. So the most complicated one is actually urine. Saliva, blood, cerebrospinal fluid, typically you can see around 40 to 60 different compounds. If you do different preparations or have higher field strengths, you can get maybe 10%, 20% more. You can also look at food and wine and other things. And again, the number of compounds is hovering around 35 to 45. So it's not thousands, but it's a good number. And in fact, all of these compounds are generally known. And so it means that it's not just identifying, it's also quantifying it's possible. When you compare NMR against other technologies, it does okay, at least when you think about both identification and quantification. So serum NMR doesn't do as well as the LCMS, but LCMS has mostly lipids, and NMR isn't very good at measuring lipid components, at least the level that LCMS can do. In cerebrospinal fluid, it's about comparable between the three methods. And in urine, actually, NMR still holds sort of the records in terms of completeness for both identification and quantification. Now, that said, each of these technologies measures a different part of the metabolome. So it's not as if these are all the same compounds. So sum these together, and you might only find about a 10% overlap between each of the methods. So again, one shouldn't be done to the exclusion of the others. And in our routine work, we always use all three technologies. Why am I talking about NMR-based metabolomics? It's because I like it. I like it because things are simple. So the KISS principle, keep it simple, stupid. NMR is actually a relatively simple process. Most of it's 1D. We don't have to do complex 3D experiments. We don't have to develop novel pulse sequences every week. Instruments are very stable. It's a very reproducible system. As a rule, operation of an NMR instrument is actually cheaper than most mass specs, again, because it doesn't break down so frequently. And the quantitation that's possible with NMR is still much better than with mass spec. And there's certainly a trend towards much more automation, which I'll talk about later on in this course. So we'll dive into some of the issues of experimental methods and protocols. And these are methods and protocols that are relevant not just to NMR, but to other fields, other technologies. And maybe we'll just start off with general concepts. So because the metabolome is sensitive to the environment and to physiology, it means that metabolite levels are affected by diet, age, time of day which samples are collected, gender when we're talking about humans and other mammals, and the body mass index. So there are very specific metabolites that can be elevated or decreased dependent on those. And so if you don't include that information in your study design, you can end up basically not measuring the metabolites that are driving a disease, but actually are driven by age or by diet. And there's been quite a few studies that have fallen flat on their face because people didn't account for these important factors. It's also important to remember that if you are collecting samples that involve biofluids or tissues, is that they're metabolically active. Blood is a living sample. There are living cells in it. There are even living cells in urine and saliva. So while it's not generally important for someone doing DNA analysis, and it's not so important for someone doing proteomics, it's very important for someone doing metabolomics to quench the metabolism. That is, kill the cells or kill the proteins. So you can do that by hitting them with rapid freezing. So if it's a tissue and you're working with surgeons or a vet or someone like that, or even a plant, freeze it quickly. And liquid nitrogen is usually the best. If you can't get that, get at least dry ice and methanol. Typically, if you can't or won't freeze things, then you can kill bacteria by adding things like sodium azide or EDTA. And again, that prevents a lot of the activity. It also means working with samples relatively quickly. Leaving things out for several hours on the bench is bad. Working in a cold room is a good idea if you can. In many cases, many NMR or any metabolomic experiments are essentially done as an afterthought. Someone's collected a lot of samples and say, hey, I've got something that's been sitting and freezing for 5 years. Do you want to look at this? And if you find out that, in fact, the samples were never quenched metabolically, or that have been frozen, thawed 100 times, or where there are different collection protocols by different labs and they're all pooled together, you won't find anything. There's going to be too much noise. So thinking about experimental design is critical, and in many cases it can save you a lot of time. And the rule of thumb I use is, a few hours of thoughtful design can save a few years of fruitless data processing. And it's just the same sort of thing as a few hours in the library can save you a few months on the bench. So there are certain protocols that have been published now for how one should handle or process different biofluids. There's a paper that we published last year on experimental methods for urine. It's a consensus paper that brought in a number of leading NMR spectroscopists who've been doing metabolomics for a while and identified issues and came up with a number of recommendations. So if you're working with urine, whether it's human or rat or koala, it's still the same thing. You still have to filter with a 22 micron filter. While it's still wet, obviously. Store the sample in liquid nitrogen or at -80. Dry ice will produce some effects, but if you don't have the choice, at least freezing is still important. Adding sodium azide or EDTA gets rid of any bacterial activity. Buffering is important. Urine can vary in pH from 4 to 10. And so pH effects can profoundly change your chemical shifts and induce all kinds of issues with peak fitting and peak analysis. When you're doing chemical shift referencing, you should only use DSS. Unfortunately, I couldn't convince everyone else to say only DSS. Frankly, TSP is a very poor choice as a chemical shift referencing reagent. It's highly pH dependent. It induces faulty carbon chemical shifts, and it's also sensitive to other problems with the matrix. You have to add a lock solvent. 5% D2O to 10% D2O is typically used. When you are collecting samples, don't group them by cohort. Randomize things. This is what everyone does, whether it's NMR or mass spec or GC or LCMS. The preferred frequency that is suggested and is advised by manufacturers at 600 megahertz, because there are many spectral reference libraries that are maintained at that one. But from my perspective, at least with urine, it's better to collect at 900 or gigahertz or 1.2 gigahertz. Again, this is something that came out of a consensus, and not all of us agreed. Working at 25 degrees and using the 1D NOSY presat pulse sequence is the consensus. It works quite well. It's maybe not the best sequence in the world, but it's how most of the reference spectra in the world have been collected, and so you need to follow that. And so there's a typical standard acquisition time, a typical standard delay, and those are the things that you have to stick with. If you don't, then you will actually have some difficulties. So this is an attempt to standardize the collection of samples. There's also been a number of other studies that were earlier and for which we drew some of our information. But there's stuff on information for serum for NMR and plasma and tissue extracts. And so these are some papers that were published. A little dated. These are 7, 8, 9 years old now. Basically from these ones, there is still a consensus. Storing plasma or serum, so it's not blood. You never analyze blood. But store them in additive-free tubes, if possible. That means you don't want to have organics like EDTA or citrate, because that makes it very, very difficult to deconvolute things with NMR. Store at minus 80, no dry ice. To get rid of the lipoproteins, which cause a lot of problems, signal broadening, you can use ultrafiltration. 3 kilodalton filters are the ones that are recommended. As a rule, they don't recommend extraction, although more recently a method developed by Dan Raftery shows that you can use a methanol extraction protocol, and it helps. Use DSS, don't use TSP. Add D2O, randomized sample orders. So the other things that we've just saw with urine are still there. So as I say, lipoproteins produce these big, large lipid signals, and to many of you, they look like distorted baselines. There was a lot of interest for trying to do elegant methods using pulse sequences like CPMG or DOSY type things to get rid of certain types of signals. CPMG or double CPMGs can be used to get rid of the lipids. DOSYs can be used to get rid of the sharper signals. They are not great. They don't produce clean spectra. They're not quantitative. Chemical removal has been consistently found to destroy a lot of compounds. The exception is being a sort of a cold methanol extraction. There's some now work doing digital removal, where now that we know what the signals of lipoproteins look like, you can subtract them out, and therefore leaving just the sharp signals. So this is a post-collection removal. It's looking promising, but not perfect. MNOVA software has some efforts in that regard. And then there's the physical removal, which is ultrafiltration, which is the recommended method, or using silica nanoparticles, which can help get rid of some portions of the protein, but not as well as ultrafiltration. So that's the biofluids. There's tissues. Again, tissues are metabolically active, so you have to quench things quickly, within minutes or even seconds if you can. In order to convert tissues into something that's a fluid, you should extract them. Water or acetonitrile is typically used. And there's some homogenization or sonication. Then you'll filter to get rid of the proteins and other particulates. And then you freeze dry the samples. So the freeze drying gets rid of the acetonitrile. Of course, it'll also get rid of some of the volatiles. But it is the method that's preferred and generally recommended. And then same sort of things. Add DSS, add D2O, collect the 1D NOSY presat with the same acquisition times and the same delays. There are methods for plants that have been published in 2007, 2010. These are some papers. And when we send these things out, you guys will be able to hunt down the specific references. But the basic essence, not unlike with tissues, is quench with liquid nitrogen, don't do any dry ice, grind the sample to powder. So this tries to essentially eliminate things that are bound to the cell walls, which are more of a problem with plants than with animal cells. Grinding is one way. Then you can freeze dry the samples if they started to turn to a bit of mush. Then you extract with boiling water or methanol water, and then filter, just like you did with the tissues, to get rid of both the proteins and other solids. Same thing, add DSS, add D2O, randomize the order, and collect with 1D NOSY presat. There's a nice picture that appeared in that, I think, the 2010 article that just sort of shows this stepwise process of taking the samples, typically freezing them quickly or adding liquid nitrogen while they're in the mortar, grinding away, transferring the powder to the tubes, keeping them in the freezer until necessary, freeze drying, and then adding out the solid material, sonicating or homogenizing, spinning things down or ultracentrifugation to get rid of the particulates or proteins, and then dropping into the NMR tube. So whether it's plants, animals, biofluids, tissues, cells, and cell extracts, there's a general consensus. So generally, 600 is the preferred frequency because most of the samples and reference spectra have been collected at that. But I think there's clear trends, certainly if you're looking at urine, to go to higher fields. There are better pulse sequences that are available, but the NOSY presat is the consensus, so we're kind of stuck with that. Two-dimensional NMR is better than one-dimensional NMR for simplifying spectra, but 2D takes longer. But things with rapid acquisition, non-uniform sampling, it's possible to collect 2D spectra very quickly. However, most people doing metabolomics still prefer 1D, and it's likely for the foreseeable future that 1D will be used. You can use eretic methods for both getting a reference standard and reference chemical shift, and also for quantification. But having DSS actually helps a lot, and again, it's something that everyone still uses, both for referencing and for quantification. If you're trying to come up with something that would improve the technology, it's always possible to come up with an amazing pulse sequence or amazing preparation protocols. But if they take 24 hours, no one's going to adopt them. So if you can keep things simple and fast and cheap, and if you can always keep that in mind, can I make it simpler? Can I make it faster? Can I make it cheaper? Those ideas will be adopted. If you make it harder, longer, and more expensive, those ideas won't be adopted. And the ironic tendency is that many of the things that are still being published as new protocols are both slower, harder, and more expensive. So no wonder most of them aren't being adopted. With respect to spectral collection, after you've collected your 1D spectrum on the spectrometer, there are some pretty standard methods, beginning with the free induction decay. Zero filling is important. It helps. Doing proper phase correction, baseline correction, solvent filtering to get rid of the water, smoothing or line broadening to reduce some of the signal or reduce the noise, getting the proper chemical shift referencing. So those are pretty much standard. There's very little difference between what labs will typically use. The only difference now in metabolomics is actually how the community will interpret and analyze the spectra. So it's nice that we've got this consensus up to the stage where we've got our spectrum. But from there, things diverge. And as I said, the difference is whether people choose to do the statistical methods, the Stockseys, Classeys, chemometric methods, or whether they choose to do deconvolution. For me, it's still shocking, but probably 2/3 of all NMR metabolomics papers use this approach, when I think even the originators feel this is outdated and have moved largely to this approach. So in that regard, deconvolution is becoming a standard, but it is a method I will strongly emphasize here. And that I think it is relevant because we now understand and can measure almost all of the compounds in a given NMR sample. We aren't dealing with dark matter. We aren't dealing with unknowns. Everything that you can see by NMR is probably a known compound in which an existing chemical standard either can be bought or easily synthesized. So once you've got your NMR spectra, then it goes back to the same thing that we saw before. It's how do we go from those spectra to the lists, whether it's lists of peaks, lists of features, lists of compounds, with either their intensities or their concentrations. So there are different tools for doing that. One tool is produced by Brooker. It's called AMIX, or AMIX. It allows you to look at both 1D and even 2D spectra, and it has a large library of 1D spectral references, reference compounds, and you can kind of match and mix those things to deconvolve. It has information about the structures of those compounds. Another very commonly used tool is the Conomics NMR suite, which actually appeared many years before AMIX, and generally is a lot easier to use. It allows you to perform spectral deconvolution. Requires a bit of experience and training. The software can be downloaded for free, and many people actually use the free version. It doesn't allow you to save anything, but you can keep on getting the download for free. It's now reduced in price and quite accessible. It has a huge library of many different frequencies, from 400 to I think 1.2 gigahertz at many different pHs. So it's got an exceptional library. Then there's some freeware, tools like Basil, which I'll talk about a little more, which does automatic deconvolution, but only for certain types of biofluids and only certain types of spectrometer frequencies. And then there's another tool called Metabominer, which is a downloadable program for doing deconvolution of two-dimensional spectra, something that we developed back in 2009. However, to do deconvolution, spectral identification, you actually need reference spectra of pure compounds. And so there's a number of spectral databases that are now available. And these have been developed over the last 10 years, and they represent very significant efforts. They're all publicly available. They're free. The Human Metabolome database has about 1,000 spectra, or spectra for about 1,000 compounds collected at 500 and 600 megahertz. There's NMR Shift DB, which has like 20,000 spectra. Most of them are not metabolites, but some are, and some of them are pretty exotic. Again, it's free. The Madison Metabolomics Consortium database and the Biomag Res Bank, which are basically one and the same, also have a very large collection of metabolites that have been collected and assigned. And in fact, the most popular downloads from the Biomag Res Bank are not the protein assignments, but actually they're metabolite assignments and spectra. Now, when you're identifying compounds, there are different levels of compound identification. And these are the 4 levels that have been described by the Metabolomics Standards Initiative, or MSI, which was started in 2006, and a paper appeared in 2007 by a number of prominent people. The best level, or level 1, is essentially something that's been formally identified. So in the case of mass spec, it means having a tandem mass spectra as well as retention time with an authentic standard. Most people in mass spectrometry never achieve this. Most people are about this level 2, where you identify a similarity to a public database like HMDB or Biomag Res Bank. You may not have an in-house standard. Now, if you can spike in an authentic standard into your mixture, then you've achieved level 1 identification according to this method. But there's also been some wide discussion within the community that if you do the proper deconvolution, using, say, Cannomics or Basal, you also have reached a level 1 standard because you're not measuring against one peak, as you are typically with mass spectra, or one retention time. You're measuring against 10, 20, 30 peaks, and you're also measuring against their intensity differences. So not only their chemical shift, but their intensity patterns have just as much information. So in that regard, people are pushing to a revision of the MSI standards, which would allow basically most NMR analyses to reach level 1. Now at level 3 is something called a putatively identified compound, and this is typical of lipidomics. So many people can identify that this is a phosphatidylcholine or PC38:3, which is fairly specific, but it doesn't identify SN1 or SN2 positions on the phosphatidylcholine. And so proper full identification would actually describe the position of where those desaturations are, the length of the individual change on the SN1 or SN2 positions, which is almost impossible. So this is really where most lipid classification identification is. And then there's the unknowns. These are peaks that are real peaks. Everyone knows they're peaks. They're reproducible, but we don't know what they are. And as I say, there's thousands of those now, and they're simply identified as unknowns. So in many publications now, people ask for you to formally declare which level you've achieved with the metabolites, with the tables that you publish, or the data that you submit. So now that people are producing lots of metabolomic data, there's obviously a need to be able to share it and also to archive it and save it. When we think about metabolomic data, we have to think of two types. One is the type with the biosamples, which represents the thousands of peaks from blood or urine or plant tissues or wine or juice. And these typically include both the assigned, and in some cases there are unassigned peaks. So we need to capture those, and then we also need to capture those reference standards, the pure compounds, the things that are in the Biomag Res Bank or HMDB. These are the ones that allow us to identify what's there and to quantify. And so in that regard, we have to have data formats that can work for both. So there have been data formats for pure compounds, but there really haven't been data formats for the mixtures, the ugly-looking data. So if we look at how other data exchange formats have evolved, we can go to structural biology. There's several well-known formats, PDB, MSIF, NMR star for biomolecular NMR, FASTA, and FASTQ formats for GenBank, UniProt. There's standard formats as well for gene expression databases, whether it's Geo or ArrayExpress. Standard for mass spec, both for proteomics and metabolomics, as a markup language called MZML. So these exist, but we didn't and haven't had one for NMR. This is just a picture of what an MZML format looks like. So it's like HTML, which is another markup format, which all your web pages are in. So about 4 years ago, a consortium was developed called COSMOS, develop an NMR standard. And out of that discussion grew NMR-ML. And so there's a paper that's being written and finalized involving several groups, including some of the ones who originated MZML, who are taking some of that. So this will replace the standard that has been used since 1987 called JCamp-DX. JCamp-DX is still used for infrared and a few other ones, but it's an ancient format, and it wasn't designed to handle mixtures or the biosample layer. Right now, the version that's being published is for one-dimensional NMR, but it's flexible enough to handle 2D and 3D, and those will be described more. MetaboLights, which is a standard repository for this, is accepting NMR-ML formats. It is now becoming part of what the Spectra are in the HMDB, so we now have NMR-ML data that's available. And you can go to the NMRML.org website, and they have all the converters and the validators. There's still some effort to try and make it compatible with NMR star, but we'll have to see. So NMR-ML looks complicated. It's not for people to write by hand. It's essentially, there are tools that basically allow you to take your spectra and just simply save as NMR-ML, or convert a Brooker or Agilent data to NMR-ML. And you can also take these data files, which include headers and detailed information about how you collected it, along with the binary that has the FID, and to view things. So it's readable. And you can also annotate those NMR-ML formats and assign chemical shifts and coupling constants and associate structures with those. So we're hoping to have the NMR-ML paper wrapped up in a couple weeks. As I say, there's a lot of effort now in the community to get the viewers, the converters, the validators, so that NMR-ML is easy to use and everyone can use it. We also need the community and people to start contributing NMR-ML formats, and so they can deposit it into the public databases, both at the biosample level and at the reference level. And many of those archival databases, which are already accepting or willing to accept NMR format, are MetaboLights and the Metabolomics Workbench. So these are the GenBanks, or the PDBs of Metabolomics. So if you publish something, the expectation is that you should put your data into either MetaboLights or the Metabolomics Workbench. MetaboLights is operated by the EBI in Hinxton in the UK, and it supports standard archival data uploads. So you can include experiments, the spectra, the lists of compounds, how you conducted the experiment, other metadata. It complies with the Metabolomics Standard Initiative, and all the data that's uploaded to the MetaboLights website is also shared with the Metabolomics Workbench as part of the COSMOS initiative. So certainly MetaboLights is one of the best places to deposit your data, and there are tools developed by Metabolomics Australia to facilitate that. A slightly simpler way of depositing data is to go through the US Metabolomics Workbench. They have a lower threshold for what's being deposited. So the deposition effort is generally a little easier. It also has a number of other tools, like a reference metabolite database, which is really just the HMDB. And it's linked to MetaboLights, the database in the EBI. So you have several choices in terms of uploading your NMR-based metabolomic data. And you can upload it currently in several different formats, but as I say, the tendency or the trend, strong push, is to try and get everyone to adopt the NMR-ML format. So that is essentially a quick outline of how you collect data for NMR. We haven't talked about the details of how you do spectral deconvolution, and we'll explore as well statistical spectroscopy. So with that, we'll wrap up,